Welcome to this Ularcirc screencast. This software was written to help researchers analyse and visualise circular and linear RNA transcripts from high throughput sequencing datasets. This screencast provides a walkthrough introduction on how to use Ularcirc for a simple but powerful bioinformatic analysis. So upon loading Ularcirc, you will see the following screen. Ularcirc starts on this Setup tab. There are a number of tabs, and each tab has a similar layout in that it has a main tab, which contains a lot of information, and a sidebar tab which provides options for that window. For the Setup tab, the main tab has quick start instructions on how to perform an analysis. And effectively, this screencast, we're going to repeat what's here. So to start an analysis, it's probably easiest to load the transcriptional database for your data. We need to load the human annotation database. We need to load the HG38 genome and the default transcriptional database. When selecting Load Transcript Database button, we are populated with a little annotation here that confirms that we have indeed loaded the reference. We are now ready to load our samples, which can be done through the Projects tab. So we're going to load a dataset called 2-Zabo. As the name implies, this project contains 2 samples, and it's from the Zabo et al. 2015 Genome Research manuscript. Upon pressing load, we are given some metadata. We're told that these datasets were aligned to HG38. The library captured the opposing strand. Click anywhere to close this metadata window. We can just double check we've got the right library prep type, and we do. We've got the opposing strand, which is the TrueSeq library. And we can see there are 2 datasets here. Now, our 2 datasets are displayed both under the Selected Sample Datasets heading, as well as under the Grouped Analysis Dataset heading. The samples that are selected in this part of the window allow us to visualize certain aspects on the Gene View tab, and we'll see that in a moment. The Grouped Analysis Dataset allows us to group samples. So we're going to prepare 2 groups, 1 sample per group. And now we're ready to do some analysis, and we do this through the Gene View tab. Before I go there, I just want to select 1 sample for visualization purposes. Okay, let's go. Okay, so we want to do a grouped analysis, and basically we're going to build a backsplice junction count table, and we want to annotate it with the parental gene. We're going to annotate the top most abundant backsplice junctions from both group 1 and group 2. And when we press the annotate button, we get we get some information that Ularcirc's doing some work here, from some status windows, generating counts and annotating. And when it's ready, it will display the table. This table contains a backsplice junction ID, which is just a genomic coordinate information. We have some backsplice junction counts for group 1, backsplice junctions for group 2, the annotated overlapping parental gene, and some what I call a RAD score, which is a ratio score, which I'll talk about in a moment. Now I want to explore some of these candidates a bit further. I want to find the most abundant circRNA in group 2 dataset. So I can sort the table by clicking the column header, and clicking it 2 times will sort the backsplice junctions from the most abundant down to the least abundant. Now I want to select this backsplice junction, and this will highlight to Ularcirc that I want some more. This is the circRNA or the backsplice junction that I want to focus on. And by doing that, and then navigating to a display gene transcripts, Ularcirc automatically selects this parental transcript for me. and we've displayed a integrative view of splice junctions. So all splice junctions, whether they're back splice junctions or canonical, are displayed via these loop graphs. So the height of the loop graph reflects the raw counts for that junction, and the position that it intersects on the x-axis defines where that junction mapped to on the genome that defined it to be a splice junction. Because we selected a column in the table a moment ago, it is highlighted as a light blue colour, and we can see there that this is a very abundant back splice junction, and there are other ones. There's a little black, another black one that has very similar coordinates to the most abundant back splice junction, and there's probably some other very minor back splice junctions scattered across this gene. The second lip graph defines our linear junctions, and these are much spliced across a much broader range across the genome, and there's quite a number of them. And you can probably see already that these align quite nicely to the annotation, which is what we see on the bottom. Our annotation confirms that this gene is transcribed from the negative strand, denoted by these arrows. So this is the 5' end. This is the 3' end. Our exons are the little solid blocks, and introns are the lines that join the solid blocks. Interestingly, if you just quickly cast your eye, you can see that annotations are not quite complete even today. As we can see, some splice junctions here that do not align to blocks below, which would infer that there's potentially an exon here. However, the majority of exons are annotated. As you sort of trace your eye across these splice junctions, they all fall across to the boundary of an exon. So it appears here that our back splice junction is actually a circularization of this exon. And we're going to zoom in, just have a little closer look. And we can zoom in by dragging these slider bars and pressing the navigate button. And here we have a better resolution of this back splice junction. Indeed, it does look quite nicely like it's overlapping this, or that this back splice junction is a circularization of this exon. So we can flip between samples pretty quickly in Ularcirc. So we're currently visualizing data from this ID. Navigating back to the Projects tab and just selecting the other sample will allow us to visualize the other ID, which ends with 284. 284. And it looks very similar to the other transcript. The counts are significantly lower though. Navigate back to the second sample again. And it should be there. There. Okay. Now our back splice junction is still selected, and I'm going to do a little bit more analysis. I'm going to retrieve the sequence for both the back splice junction and the circRNA. And this can be done through the Junction View tab. Now, Ularcirc is again telling us it's doing some calculations and some work. And when it's ready, it will display. So our back splice junction is defined by this ID. It comes with a parental gene SLC8A1. There are 12 transcript isoforms for this parental gene. And these 12 parental transcript genes have an average canonical splice junction count of 956. This is lower than that of the back splice junction, so you can kind of infer from this that this circRNA is quite abundant. Now back in the table, I quickly refer to a RAD score. This stands for a Read Alignment Distribution Score, and it's got a value of 0.5. Now, what does this mean? Basically, it is a ratio of type 2 alignments to type 3 alignments. I define a type 2 alignment as those alignments that capture the back splice junction in the forward read of a read pair. Type 3 alignments are those alignments that capture the back splice junction in the paired read of a read pair. So if the distributions if the reads are well distributed, we should get an equal proportion of type 2 to type 3 reads, and this should relate to a 0.5 value. Values that are close to 0 or 1 are probably more indicative of a false positive circRNA. We also are given an inferred length of the circRNA, and this is just a concatenation of exon sequences that fall within the boundary of a backsplice junction. And here we're given the backsplice junction sequence. The backsplice junction actually occurs at this dot. So the backsplice junction will go TGT, TAG, so forth, so forth. And if we want to recover the full circRNA sequence, we can select this checkbox here, and we can be shown that the whole sequence. We can also have a quick look to see if we can see if there's any open reading frames. So we'll click on open reading frame analysis, and we should populate with a circos plot. And there we go, there's our circos plot. And we can see that the SLC8A1 circRNA does indeed have a large open reading frame, and this is well known. This has been published back in the 90s. And this open reading frame contains 606 amino acids. So there you go. There's a quick analysis for you on a circRNAs from a heart dataset. Ularcirc has other features which I'll address in other screencasts down the track. Hope you've enjoyed viewing, and hope you enjoy using the software.